So welcome. Thank you for coming to my talk today. My name is Gavin Saari. I'm a PhD candidate at Baylor University in the Department of Environmental Science, and I'm about 3-4 months away from defending my PhD and moving on and starting my career. So I wanted to chat with you about some opportunities I've had all the way throughout my education and even taking a break kind of in between, transferring into different schools and such. So I started off at the University of Wisconsin-Superior in Wisconsin on the shores of Lake Superior, and now I'm here talking to you in Central Texas. So I'll kind of bring you through how that occurred. So for those of you, just to be familiarized, I'm from the northwest corner of Wisconsin, close to what I've circled here as the town of Superior. And it's a great area, right on the shores of Lake Superior. Grew up about 10 miles from Lake Superior, and has a bunch of different watersheds and lakes and rivers that are great to play around in, and have a great recreational opportunities to do many things. But ultimately, that's of what I kind of grew up doing, is kind of deciding that's what I wanted to do then as a career. So I'll tell you a little bit about myself, and I like to fish a lot. So whether it's on the shores of Lake Superior or some of the rivers flowing into Lake Superior, you can see I like to fish a lot. I had a family group that also liked to fish a lot. So that's kind of where some of my passions stem from, is recreationally, this is what I was used to doing. And even after I've come down to Texas, even of getting onto and fishing in the surf in Lake Superior, or excuse me, the Gulf of Mexico, but also some of these critters down here, some of these rainbow darters. We've been doing some different work actually in the Ozark Mountains on the Oklahoma-Arkansas border. And this guy up there in the hat, kind of with a big lake trout next to me, shares some of that same passion and some of the questions he's answering in his career. So I thought he'd be a great mentor to kind of foster and facilitate some of that work at Baylor. So when I was thinking of locally up in northern Wisconsin where there'd be opportunities to kind of learn and educate myself on some of these things, I went to the University of Wisconsin-Superior, and I did a Bachelor of Science in Biology and got a chemistry minor. So it's right on the shores of Lake Superior, is on the western end of Lake Superior. And ultimately, it was a great opportunity to start thinking about different fields in aquatic ecology and fisheries. So you take some of your core classes, you take biology and you take chemistry, but based on where they're at, they had a lot of emphasis in aquatics. So you can take things like limnology, where you're studying the biology in different lakes and streams and thinking about their chemical features, but also kind of their physical features at the same time. Is what makes a lake a lake? How is this lake different from the lake next door? So start kind of going through that education, picking up some of these things, and my favorite class, ichthyology, is fish, and more fish, where you're thinking about fish behavior, fish evolutionary taxonomy, and ultimately is trying to think about what is their role in these aquatic ecosystems. But at the same time, you've got to have the basis for all this in thinking about genetics, cell biology, and animal physiology and such. But you've also has to understand some of the chemistry, so different chemical structures, but ultimately different reactions that are going on in my favorite fish species and such. So it was a great place to really pick up some of these things and start an education. And actually, they had being on Lake Superior, they had a research vessel that we could actually even go run out on. They retired a tugboat, and a 58-foot tugboat that was all steel, and they equipped it out with some different research equipment, and it was used through an institute that they had there that I'll talk about in a second, but also in our classes. So we could go out in some of the harbors and Lake Superior and do some fish trawls, but also do some different things related on taking sediment cores and thinking about some of the organisms that were living in that sediment. So when I was an undergraduate for almost 3 years, I had an opportunity, saw an ad to be a laboratory technician at this Lake Superior Research Institute at the University of Wisconsin-Superior. So what they do is actually is thinking about some things related to natural resources and aquatics, and they provide research opportunities for undergraduates. And I did that for almost 3 years. And they have different expertise. They're kind of also a contracting lab, and so they do different things of analyzing whether chemicals are actually affecting some organisms and thinking about habitats and conservation of habitats, but also some invasive species that I'll kind of get to in a second. So luckily I was employed through them and working for them about 20 to 30 hours during the week, but then full-time during the summers, that I could get exposed to a bunch of things that I'll talk about actually here. So that in sciences, I cannot emphasize how much consistency matters. So whether you think about things you can have repeatable results, consistency is the basis of that. So through there, I was exposed to picking up, besides just general lab classes, but practicing those actually in an employment setting and going through a bunch of these different procedures. And to get repeatable results, you have standard operating procedures that inform you and construct you to do the same thing every single time. Every single time you're feeding an organism or measuring of anything of volumetric volume, so that you can have some sort of consistency and have repeatable results. So with that, we cultured a bunch of different organisms that I'll get to in a minute on the next slide. So we had a bunch of different organisms we cultured, and then we used those organisms to answer some different questions actually related to toxicity. And one of the first things, the test that I was exposed to, is how do we determine that our organisms are the same? We want our organisms to be just as healthy in Wisconsin as they are in Europe. So that's where those standard operating procedures come into play. But you also have to do a test to say, my fish are as healthy to a standard toxicant, like it could be sodium chloride, just table salt. In this instance, we use potassium chloride. So I can know that when we're trying to, in the broad scheme of things, when I'm trying to compare my results to somebody in Europe, we can normalize it to say, look, we're using the same organisms and they're just as healthy as your organisms in Europe. So that's a big part of it. And so I'm going to expose you to a couple of these critters that we grew. So we did some primary producers and actually cultured green algae. And what feed on the green algae is some zooplankton. And the first time I saw, these are called rotifers. So I don't know if has anybody seen a rotifer under a microscope at all? But they are the coolest things ever. And they are so cool. Actually, I've got a video to show you how cool these suckers are. And so they've got this crown of cilia to create this, let's see, how can we get this? Can we get it to play? It's thinking. So they've got this crown of cilia, and how they consume algae is create this nice huge suction. So they beat those cilia really fast. And ultimately, they beat it really fast and suck in the algae. And this structure here grinds up that algae, and that's what they consume. You can see they're transparent, so you can see their guts and mouths and everything. So they're almost some really cool organisms that, if this works, I'd like to share with you. are other types of zooplankton that have been little critters that are feeding on the algae. And these are the water fleas, and they are been shown to be very sensitive to different types of and different versions of water quality and different contaminants. So those are another form of general organisms. So these are swimming throughout the water column. But we also had some work with some of those that were on the benthic or the bottom of these different lakes and rivers. So these are some of your freshwater shrimp and an aquatic annelid worm actually, and then bloodworms, which are actually just in the same order as like mosquitoes and such. So those are really some great ones that you can use to answer some different questions of what's happening in the sediments. So ultimately, with these organisms, we were conducting a bunch of different toxicity studies. So exposing these organisms under different situations that may or may not contain different contaminants, and they were of different durations. And based on the question, if you're thinking about short-term exposure, you're thinking, do these organisms survive? Long-term exposure, do these organisms reproduce? And then ultimately, some other long-term studies, are these compounds in the sediments bioaccumulating in some of these different types of worms? And based on thinking of up the trophic level, you want to make sure that your algae's doing all right and reproducing and growing. You have to make sure, thinking of what consumes those is these types of zooplankton, what consumes the zooplankton, bait fish, and what consumes the bait fish is those big predator fish. So thinking about the entire food chain, and here we are actually doing some studies related to the base of that food chain. So some different things, other projects that we worked on is the big thing in the Great Lakes is actually invasive species. So based on the shipping industry and and coming in from different oceanic ships, they've the last I checked, and that was like 5 years ago, there was over 120 different aquatic invasive species in the Great Lakes that were brought in by these oceanic ships. And they do that by picking up ballast water for stabilization coming across the Atlantic, and then coming into the Great Lakes and actually releasing that ballast water that has contained different organisms. And those organisms can can really affect these aquatic systems from the bottom of the food chain all the way up to thinking about nutrient cycling. And so we actually did a bunch of work of testing different ways to treat that ballast water before it was discharged, so we're not trying to combat the spread of these invasive species. So we did that at the lab scale, but then actually had a big land-based site to mimic these ballast systems. And currently they're still continuing that. They've got some of these systems plumbed into on oceanic ships, and they've actually followed them all the way to Morocco, for example, to actually sample and see how effective the treatment is. Because again, you want to treat it, but you don't want to discharge it and actually have it be toxic to those organisms that are in those certain areas. So we did a lot of that, and again, some different chemical monitoring, whether different metals and such are bioaccumulating into fish tissue, wild rice, mussels, or even mosses, actually. And as I mentioned, kind of effects of these of different pollutants to these numerous aquatic organisms we were culturing, but also then thinking about habitats of actually going out and doing some plant surveys in different wetlands around Lake Superior to track and actually look and see, are there invasive emergent plant species that are also being spread and can have alterations into those wetlands surrounding Lake Superior. So from there, I graduated and was looking for a job, and across the border in actually Minnesota, there was an Environmental Protection Agency lab. So I applied there and got hired as a student contractor, and not as a federal employee, but they used some student contractors to do a lot of their studies and to create some of their data. So what a great opportunity to keep doing some different research, and as I'll talk in a bit, getting exposed to a bunch of different things. And so first of all, I wanted to chat and talk to you about the Environmental Protection Agency for a little bit. So their mission is to protect human health and the environment. And then in this specific laboratory in Duluth, Minnesota, where I was at, it's called a Mid-Continent Ecology Division, and they're thinking about the national environmental effects of different contaminants. And what they're trying to do is ultimately forecast whether the effects of pollutants are affecting the integrity of a watershed and the surrounding ecosystems. So how they do that is trying to look at the small level, thinking of the molecular level, on how different compounds can bind to different receptors or affect different enzymes and cause perturbations through biological levels of organization, thinking of molecular, subcellular, cellular, organ level, all the way up to an individual effect, as it be survival or reproduction or actually growth. But then ultimately, in ecological risk assessment, we're worried about populations. We're worried about populations of these organisms versus in humans, we're concerned about individuals. Well, in the ecological realm of things, again, we're thinking about populations. So how are different things and these different compounds affecting the population trajectory of different species? So ultimately what they're doing is linking environmental conditions to human well-being. Because if you have a healthy environment as it relates to all the things that we get as an ecosystem service, we get water, think of the food we grow and different things. We want to maintain and kind of have this one health of protecting our environment that we rely so on for water and actually food. So through there, I was involved in a project dealing with some endocrine disruptors. So these are back in like the 1990s, some scientists kind of started recognizing that some chemicals were actually disrupting the endocrine system in humans and actually wildlife. So Congress actually gone through and gave the EPA some regulatory authority to start thinking about some different things in the past, back in the 60s and 70s, about what we're discharging into water, but then also start evaluating some of those chemicals. And so what did they start doing is, based on some of this literature and some different movements, trying to screen some different compounds, and for instance, pesticides that were heavily being used for agriculture, and wondering whether these compounds had similar effect to estrogen, which is circulating through females, and different compounds to actually mimic those types of hormones. So how they started doing that is screening these different compounds, and I'm going to chat a little bit about recently a cool way they started screening these sorts of compounds, because there's about 100,000 plus chemicals in commerce that we use today, and every year about another couple thousand come out, and they need a way to make sure those are safe for humans and the environment. So what they do is actually they started doing toxicity testing in the 21st century. They started using robots. And how they're using robots is they're using different cellular assays in like well plates, whether it's 96 well plates or 48 well plates or what have you, and they're exposing cells and proteins to actually see how, if these compounds are changing the biological activity of these different molecular endpoints. So they test through a bunch of different compounds, and they're doing it in high throughput with these robots and doing kind of some red light, green light situations. Does it maybe bind to a receptor, which this receptor is responsible for a response in the reproductive pathway or different pathways related to stress. So what they wanted to actually look at and design a bunch of high throughput assays that they could get this red light, green light stuff. Do these cells grow? And think of some of the cellular and biochemical processes that were actually going on in these cells. So that was a collaboration between the Environmental Protection Agency, but also the National Institute of Health and the Food and Drug Administration. So the project then that I was involved in is using rainbow trout to actually screen for some of these endocrine disrupting chemicals to start prioritizing which ones should we be interested in, which ones are having actually biological activity, and which ones can we set aside for a little later to evaluate at a later date. But let's prioritize this. So what we did is look at endocrine disruption and to see if some of these compounds would bind to the estrogen receptor to cause different reproductive effects along that reproductive pathway. And how we did that is once we figured out if certain compounds would bind to this estrogen receptor, it's then in male to determine whether or not these compounds as a biomarker of exposure and ultimately linking it to effects, males start producing an egg yolk precursor protein, like the females do, and which females incorporate into their embryos and their eggs. But when exposed to these different compounds, males will also start producing that same protein. So we were trying a tiered assessment to try to determine whether different compounds were being endocrine disruptors in male fish and tier them and move those up the shelf and up the ladder to do more different testing with whole organisms, and this was with some actually the liver tissue. So it's very expensive to do whole organism testing, so we were doing it what's called at the in vitro level or at the tissue and subcellular level. So from that, I met my advisor actually. He was on sabbatical working in a laboratory next door from me, and he was looking at endocrine disruptors can be kind of pharmaceuticals and personal care products, and some of those actually that are in birth control pills. So he was thinking and asking some questions about pharmaceuticals and personal care products. So specifically how different compounds in the water are actually maybe affecting some fish or other aquatic organisms. So to talk a little bit now about some of the work we do in our laboratory at Baylor is ultimately more people are living in cities now than ever, and with the human population growing and approaching 10 billion by 2050, about 70% of all these people are going to be living in urban areas, according to the United Nations. So you have this concentration of people in these areas. You also have a concentration of different consumer products, and you also then has a concentration of discharge of these compounds into the surrounding environment, whether it be the terrestrial ecosystem or the aquatic system. So ultimately, if we start thinking about where these big urban areas are are at and where they're continue to be growing in the next 10 years and such, we could see North America is expected for a human population growth about 20%, but then actually if you get into Asia and Southeast Asia, it's actually expected to grow by 40%. And what we're ultimately seeing is in these urban areas, they're having opportunities to different types of medicines, and the wastewater infrastructure is not able to keep up with all those different ways of treatment to return that water back. So ultimately, the urban water cycle is become the new norm. And what I mean by that is you have your source of water, it's treated for drinking water, it gets distributed, gets used, and is collected again, goes through a wastewater treatment plant, and is then discharged back into that source. And we've figured it out that we're gonna discharge this water below of where we draw our drinking water out of. And ultimately, you have this continuous cycle where this wastewater treatment plant is discharged into a river or such, and flows downstream to then the next city or the next local town that can use it for drinking water and such. So a great example of this is actually right here in Central Texas, and actually in Dallas. So southeast of Dallas, there's this John Bunker Sands wetland, and it's over 1,000 acres, and they pull water out of the Trinity River that flows out of the DFW area, and they treat it through a big constructed wetland, and then for water reuse, once it's kind of treated, taking out some of those nutrients and such, they pump it 30 miles back upstream to a reservoir to go through that cycle again. So, and ultimately with the Trinity River, of how much water use there is, if Dallas-Fort Worth, it's projected if they don't flush their toilets, the Houston area won't have 30 to 40% of its water. So thinking about these urban water cycles are some of the things we've been asking questions about at Baylor. So in this instance of river flows are being dominated and dependent on wastewater treatment discharge. So here is it's a Texas stream or creek where the wastewater treatment plant is discharging their effluent. And as you can see, the flows away for downstream, and up above the stream, it's dry. Because at this time of the year, that wastewater treatment plant discharge is accounting for 100% of its flow, okay? So we could start thinking about these compounds that are going down the drain, and this includes pharmaceuticals and different personal care products, and we can think about where they're originating and the fate of these compounds in the environment. So they could be coming from hospitals, like I mentioned, different wastewater treatment plants. They can come from actually on-site septic systems at your own home. And then also the big thing that they've documented is actually used in different concentrated feed operations is you can have different veterinary medicines, and think of antibiotics and different steroids that they're using to grow and beef up cattle even faster. So they've actually demonstrated those different veterinary medicines flowing into streams. And there's a great paper that came out recently last year documenting about how much of this water and wastewater treatment plant effluent is being diluted in the stream. So we've kind of been thinking about these situations where it's only in those areas that are experiencing drought and are in the arid to semi-arid regions of the United States and the world. But as you can see, typically we like to have at least a 10-fold factor dilution of this water. And what you can see is in the lighter colors are where there's greater than 100-fold dilution. And then as you get to more of the darker colors, those streams are actually receiving less than that 10-fold dilution, and even less than a 2-fold dilution. And that's just not happening in the south or southwestern areas of the United States. That's happening all over the United States. So thinking about where the cities are located and thinking about this urban water cycle. So since like the mid-1990s, we've been able to document these sorts of compounds and that are present in the environment. And based on some work we've done in our laboratory, we've documented them that they've been shown to accumulate in different fish tissue. And some of these compounds are actually high blood pressure medications, antidepressants, antihistamines, so think of your birth controls or your allergy medications, but also some actually anti-seizure compounds that are shown to be very persistent. So over time, those compounds that have been demonstrated to be occurring and bioaccumulating up the food chain into these organisms, they've caused some different reproductive effects, some survival, and effects of growth on these organisms. So once you start seeing some of these compounds accumulating up the food chain, then you really start getting interested and start answering some further questions. And based on some work we've done actually in the Chesapeake Bay and the Delaware Bay on the East Coast, we can actually see that those predatory birds that are consuming fish also get the pharmaceuticals. In this instance, the osprey that are consuming fish in the Chesapeake Bay, we can actually see some of these pharmaceuticals in their tissue and blood plasma. So it's not necessarily about chasing chemicals, it's chasing the physiochemical attributes of compounds that, for instance, may make them persistent or be bioaccumulative or toxic. And so what you do is actually some different predictors of that have been associated with toxicity or bioaccumulation is this octanol to water partition coefficient. Octanol is a surrogate for biota, or like fatty tissue, and so you're trying to determine if a compound likes to be in fatty tissue or if it likes to be dissolved in water. So you think about, in the terms of pharmaceuticals, you think about some of those attributes, but also think about how fast these compounds are being eliminated from humans, and you can think about that as the same in fish. And ultimately, there's some different things of abiotic characteristics of watersheds and the water that have been able to be different predictors of whether different organisms have higher exposure to these compounds, and that would be related to the pKa, of which the pH at which 50% of that compound is in a neutral form or in an ionized form. And those compounds that are in a neutral form are kind of more nonpolar and can pass by membranes a little easier, so they'd be uptaken across the gill tissue and distributed into different tissues within the organism. So you can think about there's different metrics, and you can compare the concentration that's in that fish tissue and the concentration that's in the water, and whether those sorts of compounds are coming across their gills, so that would be that bioconcentration factor. And also then what's bioaccumulating, which would also not only be the exposure of different compounds across the gills, but also via their diet. And if it's coming through their diet, as I mentioned, up the food chain, it can biomagnify and become at really high concentrations in your top predators. So how we've demonstrated that is actually in the laboratory. So thinking of now kind of that pH story of with diphenhydramine, your common antihistamine, you could see if we look at the uptake across time of these compounds into fish blood plasma, you can actually see that at a low pH of 6.7, it's getting in the fish. But then actually if you increase the pH a single log unit, you can see quite a bit more increased uptake into the fish. And ultimately, if you increase that pH another log unit, you could see even higher concentrations of these compounds in this tissue. So thinking about the effects of organisms, again, you have to have that exposure. And then once you kind of seen that there's the exposure of these different compounds to these organisms, then you start thinking about what are the effects. And thinking in terms of pharmaceuticals, what are the pharmacological effects that are occurring in humans? And I'll talk to you about that, about the conservation of these targets actually into fish. And ultimately, again, what we see across different pH related to toxicity is at a lower pH for diphenhydramine, over time, you see that there's a higher percent of those fish that are able to survive, but then as you drop the pH, that toxicity increases quite a bit, and it really decreases their survival at the same concentration, but just across different pHs. So what they've started to do back in the early 2000s is try to predict which compounds are going to cause, be accumulating these organisms and cause some of this predicted pharmacological activity. So if you look at the, of kind of the ancestry of through life, and you can think about the conservation of these different targets through these different vertebrates. So you see humans are on the top, but there's some shared ancestry, because again, fish are also vertebrates, that you'd expect, for example, a conservation of different enzyme and receptor systems actually in these fish. So they've looked at this percent conservation in different fish species that I'm kind of demonstrating here, and they've got this different websites and like EcoDrug that you can figure out and try to start to predict the likelihood that you'd experience a similar pharmacological effect actually in these fish. And so this gets into some additional modeling of you again, when you consume a pharmaceutical, you have this internal dose circulating throughout your bloodstream. So if you'd expect an effect in this sense of fish, if the concentrations internally in their body are at those similar human therapeutic concentrations, you'd likely predict some sort of pharmacological activity that can affect their survival, growth, or reproduction. So how you do that is actually looking at a lot of the mammalian data and thinking what are some of the minimum doses in humans that cause some effects, but also maybe in different sensitivity of different humans. Maybe it can be you have to receive a maximum dose of this compound. And ultimately what you could do is actually link it to kind of thinking of that octanol to water partition coefficient of how much do these compounds like to be in water and how much of them will actually like to be in that fat tissue. And first of all, they've got to get uptaken across the gills into their blood. So whether you can, based on a water concentration, you can predict whether it will bioconcentrate or bioaccumulate into these fish at different levels. And if it reaches some of those therapeutic levels internally in the fish, then you likely predict some of those effects. So you use the physiochemical properties of different drugs in this case. And one example of really how this approach is being used is one guy in our lab previously looked at the predicted plasma concentration. So he modeled what concentration for a given water concentration would go into the fish and measured that. So they were actually pretty spot on here in the top left graph. But then also, based on the pharmacological activity of, in this sense, an antidepressant, they saw decreased binding at the drug target when they're exposed to these different compounds at these different levels of these human therapeutic doses. And if it relates that, okay, well, so what? So what changes actually in these fish is they experience the same anti-anxiety just like a human would. So in this bottom figure here, these male fathead minnows, during the day, what do you do if you're a fish? You kind of, their strategy is to hide out, hide under a rock or some instance like that, so I don't get eaten. And so during the day, they kind of are hiding out under these structures, but then during the dark, then they're out cruising around. So what they saw is that based on these same levels that are at these human therapeutic levels in fish, is that you saw this actually same anti-anxiety in which they're cruising around in the light instead of hiding out under these different structures. So the conservation of these targets are real, and it's trying to understand how much and whether or not that's occurring in the environment. So lastly, finally getting to some of the work I'm doing with is in some of the estuaries within the Texas Gulf Coast, so where freshwater is hitting the salt water. We've been doing some different assessments and looking at the occurrence of pharmaceuticals in water and then also in fish. And one that we see exceeding human therapeutic level is a specific high blood pressure medication. And so, okay, let's think about this. Is this just kind of, is just maybe the estuaries along the Texas Gulf Coast? Are these sorts of conditions actually occurring in other places of the world? So kind of to understand that sort of question and the exposure, to relate it not to just a small version of or instance in the United States, but where are these instances happening throughout the globe? And turns out they are. And so these are these class of calcium channel blockers. Is a certain type of these high blood pressure medications that act as a vasodilator and increase then in your, if you have coronary heart disease, will vasodilate your coronary arteries so you can get more blood flow, which carries oxygen and nutrients that your heart needs to keep functioning. So, so then once you start, so we've got this occurrence in these fish, so are there any sort of effects? So that's where I've had to actually dive into the, think about fish physiology and think about toxicology and ultimately pharmacology to try to answer these questions, because these are pharmacologically active compounds, and based, as I demonstrated, there's actually some conserved targets here. So what I've been doing is actually exposing different fish in the laboratory, and the top right is the larval fish. So these guys are about 4 days old, that have been shown to be the most sensitive stage in their life cycle. But also now adult fish is where you expose them to this high blood pressure medication and start to think about, does it affect their survival, their growth? In this instance, I haven't done reproduction, but I'll actually demonstrate. I'm thinking about some other endpoints related to, does it affect their cardiovascular function like it does in humans? So you follow some standard testing procedures, just as everybody can do throughout the whole world, so you can normalize to say you're doing your studies correctly. And what I've been doing is actually looking under the microscope and recording heart rates, but also looking in a different high-powered microscope to link effects on cardiac morphology and actually cardiac function, that you can actually start thinking about, do you actually have effects on behavior? So this system here on the right is we can load fish in a 24-well plate and observe their locomotor behavior under an automated system that records out nicely all of this data for us, so I don't have to sit and watch individually fish. This is all automated. And what you can actually do is, based on the different colors you see in each well, so there would be an individual fish in each well, and it tracks it across 3 different speed thresholds, and looking at the duration it travels at each speed, the distance it travels at each speed, and the number of time it reaches those speeds. So that can have some different endpoints that are related to whether the compound is actually affecting some of their normal locomotor behavior. So what I'm trying to do is then link these sorts of the exposure of these compounds to link it to the internal dose that these fish are being exposed to that would cause a similar and elicit a pharmacological effect. And in this instance, with that high-powered microscope, I can do a technique called video microscopy, and I can take videos. Thankfully, these organisms are transparent, so I can take a video of their heart. They're just a 2-chambered heart. They have a single atrium and then a single ventricle, and I can go in and actually look at, see, morphologically and functionally, does it affect their heart contractility of each chamber, like it similarly does actually in humans. It decreases their heart rate and under different mechanisms of action, and ultimately affects the number of contractions. It slows down your heart rate, so ultimately in how much blood that your heart is thus pumping. So I can go in and slow that video down to about 50 frames per second and actually do some perimeter and area measurements of each chamber when it's in contracted and then expanded, so in diastole and systole, and actually start thinking about the contractility of each chamber of the heart and actually relating to cardiac function of how much blood is actually then, is that heart being able to pump out. So I've got a little video here that actually you can see, and it looks like it's kind of slow, but so here is some just background control fish. This is the atrium pumping and expanding and contracting, and this is the ventricle. So these are kind of some of the different fun videos I get to take and actually analyze to think if it's causing some of those effects I've mentioned. And ultimately, now the last part of my research is actually being able to use larger fish. So whether it's the adult fish or juveniles of a different species, and then try to link that internal concentration to see if they get that therapeutic dose of a pharmaceutical and relate to if it affects their cardiovascular function. In this case, this is actually in a swim tunnel. So I kind of put those fish kind of on a treadmill and actually swim them to actually see if they have the same metabolic scope. Do they have the same sort of endurance when there's actually being effects occurring on their cardiovascular system or not? So I hope that I've been able to expose you to different things. I've gotten to travel about and present my work and at some societal conferences in Salt Lake City. I've actually even been able to go internationally and actually to Barcelona, Spain, to actually present some of my research. And this one of me holding the brown trout on the right was some work we did went back in Park City, Utah, just a horrible place to do some work in some of those streams, and up in the mountains, and actually look at some occurrence of pharmaceuticals in brown trout downstream from one of the wastewater treatment plants. So I've had some great opportunities to kind of get to where I'm at, and I've jumped at them, and it's been kind of great, and it's taken me many places, and there's lots of people to thank, and especially this kind of Present Your PhD program that's kind of allowed me to come and actually chat with you and share some of my cool research. So now that, I'll take your questions. Yes?
So you talked a little bit about invasive fish. I don't know about like saltwater fish, but I've heard a lot about like Asian carp and buffalo carp and common carp in the Mississippi River.
Yep.
And possibly getting into lakes. Is that true?
Yeah, so that's the thing. So that's a good question, and it's not, when it relates to invasive species, it's not about necessarily as a management strategy. It's not whether you can eradicate them, but actually keep them under control from actually changing and taking over environments. These are kind of pioneer species. They grow really fast, or have large foliage, or reproduce really fast, or something, or get really large in size, so start disrupting kind of the aquatic ecosystem. And so yeah, that's the local area, like around here, is the zebra mussels. I don't know if anybody's kind of heard about that spread, but they can change and filter nutrients once they get to a large enough level like none other. They're filter feeders. And so as it relates to invasive species, yeah, it's not necessarily about can you eradicate them, but start controlling them. So it's actually a big problem as you start changing and thinking about aquatic ecosystems. They can be major, create major impacts and actually change those different ecosystems drastically at times. Yes?
I actually have two questions. One, what was the name of that organism that you were going to play a video for us?
It's called a rotifer. So check those guys out. Yeah, and I did some toxicity studies with those, and having to use a pipette through a microscope of actually sucking them up in a pipette and then exposing them and trying to only get 5 at a time when there's, I don't know how many thousands are, in a petri dish, but you only got to put 5 in a well. It takes a lot of practice and consistency. So, yeah.
And then the other one is, with the research that you've, has there been a certain research project that you worked on that's just been like your favorite? You seem like you've done so much. Is there one that just really like you really favor?
Yeah, so that's a great question. I'm really interested in fish physiology, and as I kind of demonstrated, like fish, and I'm interested in continuing in the future post my PhD is thinking about how chemicals are affecting contaminants. So the EPA was a great opportunity to kind of think about those sorts of pathways and the reproductive pathway between the hypothalamus, pituitary, and gonad axis in those fish, and linking some effects on reproduction to those organisms was actually a great experience. And to work with some federal researchers there that had been working on that project for 12 years, it was a great experience and really jump-started me into thinking about some of those questions. Yeah.
Why exactly did you choose fish physiology first? Like, what was the first thing that really took you into the field?
So at the Lake Superior Research Institute, so they're the types of questions and toxicity studies they were doing there were kind of some long-term effects to these organisms, but a lot of them were kind of kill them and count them assays, in which does it affect their survival, and not diving into about why it was affecting that organism, molecularly all the way up to the to the individual level. And I started wanting to ask, why does these compounds in the sediment kill the fish, or in this sense some of those worms or such, of why is it doing that? So I wanted to know why, and it was a great exposure of getting to start that process for me to thinking about that. But then I just started to ask, I wanted to know why. I had to know why, and what was happening subcellularly or cellularly in these organisms that it would actually cause mortality. Any other questions? Cool. Thank you.